Okay, well, thanks for giving me the opportunity for an application talk. Usually I talk more pedantic, didactic lectures about fluorescence, so this is a little different. And those of you who know me know for years we've worked on the protein dynamin with my friend Joe, but now we have something a little different to talk about. So before I begin, I want to thank all of us in the United States. Happy Thanksgiving Day. And hopefully we'll all be having some Thanksgiving dinner later today. Okay, so first I'm talking about the protein ARC. So what is ARC? Well, ARC stands for activity regulated cytoskeletal associated protein. It's also known as ARGS 3.1. It's a key regulator of synaptic plasticity and is actually required for formation of long-term memories. So in response to neuronal activity, ARC protein level increase rapidly within minutes in dendrites, and more slowly within hours in nucleus. In dendritic spines, it promotes long-term depression and synaptic weakening by facilitating endocytosis of AMPA-type receptors. And I'll get into that in more detail. Also, in the nucleus involved, as I said, in genes transcription and other things. So remember about neurotransmission. Glutamate neurotransmission is critical for learning and memory. So we have the vesicles releasing glutamate, and these are sensed by receptors. Then I'm going to specifically focus on the AMPA receptor. Synaptic strength is controlled in part by the amount of AMPA receptor on the dendrite surface, which is, of course, controlled by the balance between AMPA exocytosis and endocytosis. So ARC reduces the synaptic strength by promoting endocytosis of the AMPA receptors in the spines. And that's illustrated here. And we might want to note that overexpression of ARC, and of course, a lot of studies have been done in mouse models, but overexpression can lead to cognitive disorders like Alzheimer's disease and Fragile X syndrome, but I'm not going to go into those parts. But in 2006, potential binding sites on ARC were found for endophyllin and dynamin. Now, since our lab with Joe Albanese and I have worked for dynamin and endophyllin for years and years, this report caught our attention. In fact, the structure of the C-terminal domain of ARC was solved by X-ray crystallography and found to be almost superimposable on the structure of the HIV GAG domain, which is shown up here. And we'll get more on this later because that turns out to be very interesting. But let me refresh your memory on what dynamins do. Dynamins are about 100 kilodalton GTPase pinchases that catalyze vesicle budding. So you get a vesicle forming at a, for example, here we show a clathrin-coated pit, and the dynamin is important to, when it hydrolyzes, to clip off the vesicle. Okay. So our first entry into this field was, we showed with Joe's group, and Chris Beyer was the grad student involved here, that ARC definitely enhanced dynamin polymerization and GTPase activity. And this was very dramatic, and we thought it was interesting because you're seeing the consequence, not just putative binding, but that ARC really does affect dynamin 2 in particular at physiological ionic strength. But then later, two papers came out that changed the way we think about ARC. Namely that, again, going back to this retrovirus-like GAG protein domain, that this was involved in, it was reminiscent of, remember, the GAG protein is the famous viral protein, and this had some viral properties. So specifically, we did some early electron microscopy, negative staining on the left, and we saw these particles, okay, which we certainly never looked at as potential virus. But then Shepard's group later on did cryo-EM, and they identified a lot of beautiful particles that they correlated very well, they thought, with virus type particles. And the idea was that Shepard and others have, that ARC oligomerizes into virus-like particles that contain ARC messenger RNA. These can be released from the host plasma membrane as extracellular vesicles and then internalized into neighboring cells. And Shepard showed this by actually growing cells expressing ARC and collecting the medium, not the cells, but just what was in the medium, and then introducing that to cells that absolutely did not have ARC in them, and then they then started expressing ARC. So this mechanism seems to be real. In fact, it really caught the attention of the popular press for a while as articles appeared about this, how brains, now, of course, this is the popular press view, share information. This even was in Wall Street Journal, had an article on it. So Shepard got a lot of popular press on these ideas. Now, we had showed with Joe's lab that ARC can be palmitoylated, okay, which is obviously important when you think about something localizing to membranes. And here on the right, we see the structural domains of ARC. Again, you have that capsid-like, gag-like domain on the right. But I want to also point out we have the dynamin binding domain, the endophyllin domain, AP2 binding domain, which is another endocytic protein. And because of Belen's talk earlier, I want to point out that there's a presenilin 1 domain 2, and that's the enzyme, of course, responsible for clipping the APP into the A beta peptides that she talked about. So there's even a connection to that. Now, we first show that ARC binds directly to phospholipid vesicles, okay, and we can see binding curves, just pull down assays that it binds stronger to false lipid preparations, total brain extracts, but also to pure pure PC vesicles. So then we, of course, thought to do GUV work and began to explore this question with Leonel when he was still at the LFD using the famous GUV systems. And here you see our first image, and for those of you that know GUVs, you can even see these are when we had it bound to the platinum wire. And we labeled ARC covalently with Alexa 594. And I want to add that I think Leonel was very impressed because he had never labeled a protein before. But at any rate, we went through that, and that was fun. And you can definitely see on the cell surface from this and many images that ARC would bind to it. But then the next thing, when Leonel started to do GUVs with Lordan with ARC, that was really the big thing for us. So Leonel got this picture of a GUV labeled with Lordan after addition of ARC, and I think it's just beautiful. You see all these internal vesicles, okay? And moreover, because he did this on the Zeiss with the spectral imaging, you could see that the internal vesicles, the Lordan is in a more fluid state than on the outside one, on the main GUV. So that was really something. And I think that image, and that image alone, helped Joe Albanese and I to get an NIH grant. Then we had Nick Heddy started collaborating with us. So here's some more of the work that Leonel did. Again, many cases, you see these beautiful internal vesicles. And when you look at where the ARC labeled with Alexa is, you can see that it's inside in the vesicles. So that was interesting. But then Nick continued this GUV work, and he did a lot of vesicles. And here you see DOPC with Lordan without the ARC, and when you add the ARC, and he studied the time course, et cetera, you see formation of vesicles again and again. But he did a lot of studies to actually get some statistics. So you can see without the ARC, you only have a few that show some internal vesicles, not those nice, beautiful ones we saw before either. But when you have ARC or by itself or Alexa labeled ARC, both cases get a tremendous increase in the number of internal GUVs with internal vesicles. And we had different controls. We even put in EGFP to show that another random protein doesn't cause this. I think we had some other ones too. But at any rate, this is a real phenomena. And we just recently published a paper on this, membrane remodeling. So that just came out. So our current thinking is about ARC, R meaning, I guess, the field. ARC is involved in processes that require either positive curvature, this would be the AMPA receptor endocytosis, or negative curvature, which is like the release from the membrane, like the GUV case. And it's interesting to note that in the AMPA receptor endocytosis, you need the whole endocytic machinery, the dynamin, endophyllin, AP2, et cetera, et cetera. But in the release of the vesicle release, like with the GUV, ARC can do it by itself. And there's other cases of some viruses that can do that to cause release of those negative curvature vesicles. So in living cells, as I said, in the synapse, ARC facilitates endocytosis of AMPA receptors. In the nucleus, it regulates transcription of genes involved in learning and memory. So we were interested in both in the cytoplasm and the nucleus. So we started to do the usual array of nanoimaging methodologies available at the LFD. Here we looked at the RICs for ARC in the cytoplasm and the nucleus, and you can see it definitely was more mobile in the nucleus than in the cytoplasm. It's fairly slow in the cytoplasm. Presumably it's binding to other proteins or structures like small vesicles. We need to explore that some more. But in the nucleus, we were interested, what other differences are there? Well, we looked using N and B, and this is great in this workshop. I don't have to discuss how these methods work, okay? You all know that now. So in N and B, you could see that, especially in the cytoplasm, you could get higher order oligomers. In the nucleus, it was much more leaning towards monomeric states. But we follow that up with FRET, and again, here's the famous phasor diagram for FRET between EGF. We expressed EGFP alone, but also then EGFP ARC with ARC-M cherry. And you can see by that phasor diagram, there's definitely energy transfer. You all recognize that signature now. But interestingly, when you look at the energy transfer, you see the EGFP is throughout the cell, but the energy transfer occurs mainly in the cytoplasm, very, very little, if any, in the nucleus. And that leads us, again, to believe that ARC is more monomeric in the nucleus. And again, this is still a work in progress. We need to explore that, okay? Now, interestingly, a major pool of ARC associates with promyelocytic leukemia nuclear bodies, PML nuclear bodies, which you may remember face separated membraneless organelles involved in, among other functions, epigenetic control of gene transcription, okay, in the nucleus. And people had seen this a while back here with 2007 paper where they had ARC-YFP, and then the PML, they had fluorescently labeled antibodies to that. But Nick Hetty did this more recently with PML labeled with EGFP and ARC-M cherry, and you could see signals, of course, of both of these in the PML nuclear bodies. And then Nick did some FRAP analysis, which was kind of fun, I thought. And here you can see on the left where he the different PML nuclear bodies that he frapped. And here on the C, we see the pre-bleaching, and then when he bleaches it, it's a very, very, very slow recovery of the PML EGFP. However, the M cherry recovers very quickly. I mean, you can see the difference in time, between the recovery of the M cherry and the PML, which tells you that the M cherry can exchange out into the in and out of the nuclear body, but the PML EGFP is much slower. We then wanted to see if we could see barriers to the motion of ARC, and we did the famous two-dimensional spatial pair correlation function. So you see virtually everything we talked about in the early part of the course, we did this. And this, of course, was done again by Nick Hetty at the LFD. And it was quite clear that M cherry inside the nucleus had very few barriers. It was pretty much isotropic diffusion. But outside, there really were barriers, okay? And the anisotropy of the motion outside was very distinct. And you remember you're seeing those little lines there showing barriers of diffusion. So we think that inside, it's free to go around the nuclear body, but when it's outside, it's finding obstacles to diffusion, presumably things perhaps to do with the heterochromatin structures, okay? So Obviously, this too, we'd like to explore some more, but you can see the utility of these methods to explore these type of questions. Now, I also said that ARC and Dynamin cooperate to regulate actin cytoskeleton and dendritic spines, and I really don't want to spend the time to go into this. You can see with ARC is enriched and actually causes a formation of filopodia. We have some movies, but I don't have them here. But you can even see actin whiskers, and these are studies Nick did with LifeAct, which you know is a peptide that binds to actin, has a GFP attached to it. And you definitely see more of these little whiskers resemble actin whiskers, which are like thin filopodial dendrites on the ARC-treated cells. And I mean, you just have to take my word for it. When you do a lot of them, here's where we maybe need Federico's machine learning programs. You can really see that these form. So that again needs to be studied more. So again, this relates to the possible mechanism for intracellular ARC transport, not just releasing the vesicles to the outside, but maybe tunneling through the filopodia. Because in here, you do get ARC is in the filopodia. Okay, so that suggests this sort of possibility. Again, something to explore. So obviously we have a lot more to do. This is still early days. We know that ARC olimerization, we've characterized a lot in vitro, but stimulus-dependent changes in oligomerization of ARC in neurons or neuron-like cells remains to be explored. Mechanisms of nuclear transport in association with nuclear binding partners needs to be explored. So fortunately, there's a lot we can do. Now we just need to get another grant, which we keep applying for.